Good morning, students. Today's class, we are going to study about the bioelectric signals. So, in that, we are going to focus on the three different types of signals: ENG, which is called electroneurogram, ERG, which is electroretinogram, and EEG signal characteristics. So, if you see electroneurogram, it is the study of electrical activity of the nerves. And here we can able to measure the nerve conduction velocity using this ENG. And if you see, we can able to record this ENG signal by using a concentric needle electrodes by inserting into the skin or in the surface by placing the electrodes in the surface. We can able to measure the ENG. So in that silver chloride electrodes can be used. It may be a metal surface electrodes or a metal gap electrodes based. So we can keep it on the surface and measure the signal. And one of the things which we can able to measure in this ENG is mainly how whether the nerve is conducting properly or not. So in case of few patients like stroke patients or paralysis patients, so definitely the nerves will not be conducted. So for those kind of patients, so how to check the nerve conduction velocity is proper or not. So I think in a biomedical instrumentation lab, if you're a biomedical students, you might have done an experiment. That is, you will be having a set of ring electrodes kind of thing, which will be placed over the finger, and another electrodes like a surface electrodes, which is placed above the elbow or the wrist. And then the lead wires from the electrodes will be connected to the what you called an ENG amplifier, and then you will be applying a short current in terms of milliamps into the electrode. So when the current is passed to the electrodes, what will happen? The current will be passing from this the ring electrodes, which is placed in the inner finger till the wrist, because another electrodes in the wrist. So when the current is passing here, so it will take some time. So when you're giving the stimulus, it you can able to feel the shock. When the current is passed, you can able to feel the shock. So in that, the distance between the index finger, that is the finger, index finger and the wrist, which will be considered, which will be measured, multiplied by the time which is taken for giving the stimulus and getting the response, you can able to measure the time period. So using that, we can able to calculate the nerve conduction velocity as the time into distance. So we'll be getting the nerve conduction velocity. So for a normal people, the nerve conduction velocity will be normal. But in the case of people who are affected with the stroke or any paralysis, definitely the nerve conduction velocity will be less. So generally, when you see the ENG signal amplitude is in the order of 10 in terms of microvolts, and the duration will be 100 to 300 microseconds durations, and the start stimulus will be applied in the form of pulse about 100-volt amplitude. And here sometimes the instrument is susceptible to power line interference and any noise, so that we can eliminate it through some filters. So now in this figure, you can able to see that the nerve conduction velocity, which is measured in the wrist region, above elbow region and below elbow region. So here there are three different traces of nerve electron, that is nerve conduction velocity is here. It is present. Okay. So here in this three traces, there is some latency period is there. Latency means the time which is taken for giving the stimulus and getting the response, that is latency period. So if you measure the nerve conduction velocity in different regions like wrist, elbow and below elbow, it is B elbow, above elbow is A elbow. So you can able to see there is some uh, time lagging latency is there in each and every waveform, which is measured in the wrist, below elbow and above elbow. And here you will be placing the surface electrodes to measure the ENG signal. So the signal is amplified with a gain of 2000, and it is filtered with the bandwidth filtered to the bandwidth of 10 hertz to 10,000 hertz. Now you see the typical nerve conduction velocity. So generally For the nerve fibers, it is around 45 to 70 meter per second. And these are the general values, normal values. For heart muscle, it is 0.2 to 0.4 meter per second. And for in the case of when there is a time delay exists between the atria and the ventricles, that time it is 0.03 to 0.05 meter per second. And in the case of any neural disease, definitely the nerve conduction velocity will decrease. And the next signal is about the electroretinogram. So which is nothing but the electrical activity which is generated in the retina of the eye. We call it as electroretinogram. And this ERG signal, it is used in ophthalmological research as well as in clinical use. In both purposes, this ERG signal is used. When you say the research purpose, by using the implanted microelectrodes. This microelectrodes is placed in the retina, and the reference electrodes, we call it as an indifference electrodes, is placed on the surface to record this ERG signal. And in the case of clinical use, if you see, they will be placing a cornea electrodes, which is made up of a small special contact lens, and a reference electrodes which is placed on the earlobe or forehead. And if you see the ERG, there are four components, four waveforms are there: A negative, B positive, C positive, and D negative. And you see A and C waves are contributed by part of cornea. And the B wave, it originates from the bipolar cell region. D wave associated with the termination of stimulus. And if you see the voltage levels of the ERG, it is 0.5 microvolts to 1 millivolts, and the bandwidth is around 0.2 to 200 hertz. And this is a typical ERG waveform. So you can able to view that A wave and A wave is at the originated at the bottom, and a B wave is at the top. So this is called a biphasic waveform of the ERG for a normal patient. And if you see this ERG waveform, you can able to see the A wave originate at the this bottom point and B wave at the top. So here we are measuring the amplitude from the baseline to the negative trough of A, and then from A to B we are measuring the amplitude. And then the second one is like a time from a flash onset from the bottom trough of the A wave to the peak of the B wave. So here we are measuring the time which is referred to as implicit times. In ERG, we measure this time as implicit times. So this is the different layers of the retina in which the A, B, C and D wave alternates. So this is the different layers of retina. So if you see, this is the topmost and pigment epithelium, and A wave, these are the rods and then cones. So A wave is associated at this point, cones, and the C wave is present in this point like pigment epithelium, and B wave associated with the bipolar cells, and the D wave is at the end of the stimulus of bipolar cells, and these are amacrine cells, etc. So these are in these places only the waveform originates. Then, so to record the ERG signals, we are using different types of electrodes. So you can able to see the different types of electrodes here. One is the Burian speculum type electrodes. This is the Burian speculum type electrode, and this is like a cotton wig electrodes. So this will be placed over the eye to measure the retinal potential. And these are the different types of electrodes which is used in ERG. One is the jet electrodes. This is a gold plated electrode which is very thin. And this is a skin electrode. And this is the DTL electrodes. And this is like HK loop electrodes. And this is Burian Allen electrodes. So if you see the record the ERG potential which is in the microvolts range. So for different electrodes they measured the ERG potentials. So the best one is by using Hansen Burian electrodes, you are getting the better potential amplified waveform compared to the other one like universal jet, Darson filaments, and skin electrodes. If you see that amplitude is very less compared to the other electrodes which is used for recording the ERG signal. And if you see the clinical use of ERG, it is used for evaluation of the visual function both in infants and children. And we can able to determine the presence or absence of the retinal functions. If there is any retinal degenerations, we can evaluate the progressions and confirm the diagnosis of any dystrophies, and any toxic retinopathies can be detected. And also this ERG is used in diagnosing the retinal conditions in which don't match with the visual complaints. And what are the factors which affects the ERG? There are three different factors: physiological, instrumental, and artifacts. Physiological factors, if you see, the pupil, movement, age, sex, error, diurnal variations, dark adaptations, anesthesia, this will affect the ERG signal. Instrumentation in the sense the amplifications, gain, stimulus, and the electrodes. Artifacts in the sense when the eye is blinking continuously, when the tears is being found, eye movements, these affects the ERG signal. Sometimes the air bubbles under the electrodes will affect the ERG signal. And the next signal is about the EEG signal. So EEG, we call it as a brain waves, represents the electrical activity of the brain. The main parts of the brain, when you see, it's the cerebrum, cerebellum, brainstem, which includes brain pons, medulla oblongata, reticular formation, thalamus, etc. So in this cerebrum, it divides into two hemispheres, and it has been separated by a longitudinal fissure called as corpus callosum. And outer surface of cerebral hemisphere, we call it as cerebral cortex. It composed of gray matter, the neurons, which is gray matter, and separated into the regions by fissures, I'll say. And beneath that gray matter, we have that white matter, the nerve fibers that leads to other parts of the brain. And if you see the cortical potentials, it is generated due to the excitatory and inhibitory postsynaptic potentials. So these exhibitory and inhibitory synapses are developed by the cell bodies and dendrites of the pyramidal neurons. Excitatory in the sense when there is a stimulus is there, the cell bodies and dendrites will conduct the signal. And inhibitory postsynaptic potential means there is no conduction of neurons will take place. And when there is external stimulus is given, it's generated in the signals and it corresponds to the brain signals. It will be recorded as in the scalp using the surface electrodes. And if you see the EEG signal, there is a standard international, according to International Federation Society for EEG. So, and clinical neurophysiology, they recommended 10 to 20 electrode system placement for clinical EEG recording. So, in that, why the name called 10 to 20 electrode system is from the nason to inion, the distance between the different at a distance between a different lobes, it is 10 to 20 distance. That's why the name is given like that. Now in the EEG signal, in the instrumentation point of view, it can able to record at 100 microvolts per centimeter with 30 mm per 8 for 10 to 20 minutes over 8 to 16 simultaneous channel. It can able to measure the EEG signal. And using the needle electrodes, we can able to record the EEG signal. And also using electrocardiogram method, in which the electrodes will be placed over the exposed post of cortex and measure the EEG. And this is the 10 to electrode 20 system, 10 to 20 system of electrode placement. This is nason and this one is inion. So in between this is frontoparietal, and this is frontal region, this is central region, this is parietal region, this is occipital regions, and this is prefrontal regions. So here the distance between nason to prefrontal region is 10, and the frontal to that is these two region is 10%, and from frontal to central is 10%, and this one is central to parietal is 20, and parietal to occipital is 10. So like that it is 10, 20, 20, 20, and 10. In the combination, so the distance is there. And if you see the commonly frequencies of EEG are 5. One is delta 0.5 to 4 Hertz, theta 4 to 8 Hertz, alpha is 8 to 13 Hertz, beta is 13 to 22 Hertz, and gamma is greater than 22 Hertz. And when you see the alpha, it is like a resting state of the brain, and theta is like a semi-sleep state, delta is a deep sleep state, and beta is a relaxed state, gamma is an excited state. And when there is a focal brain injury and tumors, it leads to abnormal slow waves in the corresponding region. So you can able to see the first one is the delta rhythm, which is the lowest frequency, as well as this refers to a deep sleep state in the patient. And the second waveform is the theta waves, theta rhythm, so which is the semi-sleep state. And C is an alpha, which is the person is in a relaxed state. D is a beta, in which the person is in alert state. And when you see this, the last one E waveform, when the eyes is open and shut, so that time the EEG is recorded. So these are the different types, waveforms of the EEG. So thank you all of you for listening.